gratitude to COC and KSOS for this rare and unique opportunity. More than meets the eye, the role of gut microbiome in systemic and ocular disease. Let us just take a couple of deep breaths and ask ourselves a few questions, because science thrives through questions. The first question is always, how much do we really know? In fact, most of what we know is what other people told us. So logically it follows that any errors in these other people's knowledge would naturally get downloaded into our brain. So then the natural derivative is a significant part of our knowledge is wrong. And science is the method of fixing it, step by step. I'll take you back 300 years ago. Some of you may have heard of this so-called vegetable lamp of Tartary. Travelers such as Sir John Mandeville from 1357, who traveled to Europe, came back and said there was a plant growing in Europe. Instead of fruits, it produced lamb. I mean the animal lamb. The story stuck. Many other travelers also went there and they also came back with these stories. Yes, there was a plant that actually produced lamb that was edible as part of its fruit. It took 300 years of research to find that this was fake. There was no such plant, as we know, but the belief persisted several centuries. Now this is 1357, and we are now in 2026, and we are hearing tall stories about many things, among which gut microbiome, gut bacteria, are dominating social media, YouTube channels, and some conferences. There are claimed links to many of these diseases. The list keeps getting longer, and you just have to turn on your YouTube to see many people talk authoritatively about why these gut bacteria are causing everything known to man and how they can fix it. Patients liberally self-medicate. It's a multi-billion-dollar industry worldwide. They make more money than all the doctors put together, I guess, selling probiotics. Doctors not falling behind, they prescribe liberally. So with that intro, let me take you through a story. I call it Angry Cows. It's a farm story. Just take a look at this picture. The picture of peas. This view is from a farm. You can see the fence over here. That's the fence of the farm. You can see various cows grazing peacefully. The lush green grass. You can almost sense the level of peace in this picture. The cows are happy. And now, after a particularly bad summer, the grass dried up. So the cows had difficulty. In the past, some of these cows would come and eat some of these leaves of the fence. And because of the natural trimming, the fence would regrow. That is what it used to be. But now, all the cows, because there is no grass, started and they came and attacked the fence, desperate for food. And soon the fence was breached and they entered the farm. They created havoc, destroyed all the produce. See the roosters fleeing. Finally, the farmers had to come out with sticks and drive the cows away. That is the end of the story. take you through the anatomy of the small and large intestine. We got a stomach. The blue part is the small intestine, and the brown is the colon or the large intestine. The colon primarily it absorbs water and it ferments fiber. It only does two things that we know of. You can see as we progress from the small intestine to the large intestine, oxygen gradient. You see, the small intestine gets a lot of oxygen, but the concentration of oxygen is lower in the large intestine. So naturally, oxygen-feeding bacteria or oxygen-tolerant bacteria live in the small intestine, and oxygen-intolerant bacteria live in the colon. These bacteria cannot take oxygen at all. If you show them any oxygen, they will die. So the colonic bacteria do not enjoy the presence of oxygen. That's why they are anaerobes. So that is a background of anatomy and some basic physiology here. Let's look at the colon part. Because when we say gut bacteria, for the most part, we refer to colonic bacteria, not the small intestine. Now here's a microscopy of the colon. Here's the mucosal lining, and that is the lumen. The top part is the lumen of the colon, and here is the microscopy. You can see the goblet cell. The goblet cell produces mucus, and the mucus is green in this picture. I hope you can appreciate the green for mucus. And the mucus has two layers. You got outer and inner layers. So the goblet cells, see that? They make the mucus, and the mucus keeps going in this direction, and that's the lumen. And there are some bacteria here that feed a little bit on this mucus. And then there are these bad bacteria that live in the lumen. They're like outside the fence. They're not allowed inside the fence. They do their business in the lumen, whereas these commensal bacteria are living within the mucus layer. So this is the peaceful situation in the colon. Here's a farm, here's a mucus, healthy bacteria, and the luminal bacteria. And now I'll show you the picture. To the left is a healthy gut. Here is the intestinal lining. Here is the mucus. And here are the bacteria in the lumen. And here are our resting immune response. All the T cells and the B cells happily sitting there because it is times of peace. But in a damaged gut, see what happens? These harmful bacteria, they breach the layer. They enter the walls of the intestine, provoke these resting cells, and they all become angry. So that is inflammation. It does not stop at the lumen. They travel to different parts of the body, such as the heart, the liver, the brain, and even the eye. So now, this picture is called dysbiosis, when the balance of the microbiome was destroyed, so it's linked with all these chronic diseases. As I mentioned, it can be linked to metabolic syndrome and these other conditions. So now let's go through this story just one more time, really quickly. This is the mucus layer, and the farm is the colonic mucosa. It is our farm. So our farm is guarded by this fence, and these bacteria are just living outside. They are free to graze a little bit of the mucus, but not too much. And they're feeding on the grass, which is basically the fiber that we eat in our diet. The moment we stop eating fiber, there is no food for these bacteria. So what they do is, they come and attack the fence, and they kill the mucous membrane, kill the mucus layer. They enter the farm, which means these bacteria are entering the colonic wall, provoking inflammation. So these are our T cells and our B cells trying to throw these cows out of the farm and restore peace. That's inflammation for you. So this is a story you can tell your children and your near and dear to explain why they need to eat their veggies. As a child, I was never fond of veggies, but now I think I can eat a little bit more after reading this. And more is coming up. So, gut mucus, I know it's an icky term, but mucus is part of our protection. How to protect gut mucus? Eat these legumes, plant fiber every day, fermented foods with resistant starch. They are good for the gut mucus. And among them, I have singled out number 3, which really I used to look at it with great disdain whenever I found it on my lunch plate, vareca. It was always last priority for me. But now my respect for vareca has gone up. I'm sure yours too will be at the end of this lecture. So the advantage of these things are they contain certain ingredients that will reach the colon and feed the colon. They'll feed the cows. So this is the food for all the cows that live in our intestine. So it keep the cows happy, they will not come and damage your intestine. So that is your vareca thoran for you. And another popular dish which we often buy at home is kurkugalan, which has enormous pieces of raw banana in it. So they are a rich source of resistant starch. So those are food for the gut bacteria. Now, what is the difference between the rice we eat? We are about to eat lunch here. What's the difference between that rice and the paranganji that many people eat the next day? The rice we eat gets digested in the duodenum and the small intestine, and it is fully absorbed. But the paranganji, because it's kept overnight at a low temperature, the starch is altered so much that the small intestine is not able to digest it. That's why it's called resistant starch. So it goes past the small intestine and reaches the colon, where the happy bacteria chew it up. So to put this in simple terms, kanji for the small intestine, which is made of rice, and paranganji for the large intestine. So that's a good way to remember that. Kanji basically is freshly boiled rice, which you can eat today afternoon and the evening. It is still regular starch, but if you ferment it overnight, it cannot be digested by the small intestine. So it becomes food for the large intestine. Now let's go through how to destroy it. We talked about how to construct it, how to destroy gut mucus. If you do those 3 things, you can easily destroy a gut mucus and make yourself sick in the process. I love my junk food, but I do not endorse eating it every day. Now let me take you through a mucus destroying experiment. It's called the Tim Spector McDonald's experiments, the real McDonald's. Now this happened in Dr. Thomas Aaron Verghese' UK town. He lived in a town called Aberystwyth, right? As I understand, right? So that's where he lived. So this experiment occurred in that area. Now, you might have seen Professor Tim Spector. He's a renowned scientist. He has published so many papers on multiple topics, including COVID. Now, his son was studying in the University of Aberystwyth. So he made a deal with his son. He would not give his son much money. He would just give him just enough for the day because his son didn't have any money. So there was a little bit of friction there. So he said, look, he said to his son, no, I'm going to give you a project. If you do this for the next 10 days, I will sponsor all your food. Son said, fine. Because son was in university, he was starving for money and food. So professor said, I'm going to make a deal with you. You got to eat at McDonald's for 10 days. Nothing else. Son said deal because he was craving for it. He had a craving for all this French fries and other good stuff. So this is what it looked like. This is Tom, who's the son of Tim. That's him at McDonald's. He went on McDonald's for 10 days. Do you know what happened? The first 3 days he was really happy because he was getting all the junk food he wanted. But by day 4, he wanted out. He just could not take it. So Tim, his father, was also testing the son's gut bacteria, and he demonstrated that the diversity of gut bacteria, the variety of gut bacteria, diminished by 40% in just 3 days' time. Because he was not eating any fiber, the fiber fermenting bacteria went away. The others took over. That's called dysbiosis. SCFA is short-chain fatty acids, predominantly butyric acid, which is a fuel for the colon cells. There was no fuel for his colon cells. He felt weak, washed out. Even his friends started asking what happened. So then finally, after 10 days, he rushed back to eat his salads and he was happy. The story had a happy ending. But Tom proved his point. If you eat ultra-processed food continuously and avoid plant fiber, you will kill your gut bacteria. No antibiotics needed. Let's look at the story of our own gut bacteria. When we were born, we were like a phone with no apps in it. It's completely empty. No bacteria at all. So the apps were slowly downloaded onto the phone as we grew. And by the time we reached about 5, we had reached nearly what we have today. So the climb in the gut flora occurred rapidly in the first year, and then a little bit till year 5, and after that it stabilized. This picture is self-explanatory. I'll walk you through it. From birth to 3 years, for the first 6 months, breast milk gives them bifidobacteria. And then after that, the baby starts to be weaned. Dietary diversity also produces bacterial diversity in the gut. So weaning equals diversity. And for those who have an interest in gut bacteria, I'll encourage you to read what is called the twin studies. What is twin study? A twin study is unique because you're finding 2 people who share the exact same DNA. How fascinating is that? So you're removing a lot of variables. When you do the study, you're removing a lot of the conventional variables. You can't say it's in the gene, it's because of the family. No. You're looking at 2 genetically identical individuals. And a lot of work has been done with the gut microbiome and that. I'll just give you the highlights because we don't have a lot of time to go through all the studies. So number 1, what the baby eats or what the child eats decides the microbiome, not the genes. And if one of these became obese, their gut invariably had a deficiency of fiber fermenters, the cows that I showed you in the beginning. The bacteria that chew on plant fiber, they were diminished in obese twins. Very important finding. The duration of breastfeeding decides the long-term composition more than the diet. And of course, we know antibiotics destroy everything. And there's a lot of talk about cesarean versus natural delivery. Really, there is not much of a difference. Theoretically, there is, but it's not enough to make a real-life difference. So, to put this whole slide in one sentence, breastfeeding and diet, along with antibiotics, are the chief variables that decide our microbiome from when we were born. So if each one of us wonder what kind of bacteria live in our system, these 3 variables decided it. Now this is a big question. Is obesity linked with gut bacteria? If you look at literature, there are some trends of certain bacteria being more, others being less. Without reading the whole thing, you get the picture. Low diversity is the key. People who are obese or people who have obesity have less diverse bacteria. Gut bacteria, you would have heard talks about them being a metabolic organ and so on, immune regulation. But here's the interesting part. Suppose a person starves, suppose we don't eat any food. These bacteria can step in and make our digestive enzymes more efficient. So in other words, they can make you absorb more food in times of starvation. That explains why some people gain weight even if they eat very little. I've heard this so many times in my clinic. Doctor, I eat very little, but I still gain weight. One of the reasons is this. They have the wrong gut bacteria. Here is a great study. They looked at gut bacteria. They took gut bacteria from obese human beings and put them in germ-free mice. That is, mice without bacteria. If you put, can you see the picture here? From the obese person, if you give obese bacteria into a mouse, it grows into an obese mouse. But using the same food, if you give the lean person's bacteria into a mouse, it becomes a lean mouse. Remember, both these mice were given the same food. The only difference was they were germ-free mice, that is, they didn't have bacteria, but we gave them the bacteria. The bacteria were installed in the mouse. So these are landmark experiments done by these individuals. That is Professor Ridora and Professor Gordon, who have some of the landmark papers in gut microbiome. This is a very long story, but I've given you just a summary. So in other words, this is proof that the bacteria living in an obese person can produce obesity in a mouse in spite of eating a fairly normal diet. So here's the question: should we be prescribing probiotics? That's on everyone's minds, right? So I'll say this. Never blindly believe any research conclusion. I've written on this. You could look up my article. It's keyword, how not to be fooled by reading research studies, because many papers will say many things. It doesn't mean that they're all right. So I've decoded how to read these scientific papers without wasting time. And to understand papers very well, we really need to understand epidemiology, immunology, so a bit of physics, biostatistics, pharmacology, medical ethics, behavioral science, and all these other things like genetics, virology, immunology, everything. If we know all of these topics together, we'll be able to make better sense of scientific research studies. So these social media pundits have none of that. All they read is on Google, they will see a conclusion and they copy and paste the conclusion. That is why we're getting a lot of misinformation. I'll encourage you to read Nicholas Taleb, Nassim Nicholas Taleb's book. Many of you have come across this book. It's called Fooled by Randomness. Now, this is the man who predicted the 2008 stock market crash worldwide. It is considered a black swan event. He wrote a book on that. It's called The Black Swan. And this is a follow-up to Black Swan. Basically, what he says is, we see a lot of successful people here, right? And many of us think that it is because of our ability that we became successful. Nassim Nicholas Taleb says, many of our success was by random chance, not by sustained effort. So he says that human beings underestimate the power of random events in their life, and they overestimate their ability or their academic brilliance or abilities. So that's why he says it is fooled by randomness. He uses the picture of a black cat. Remember, if a black cat crosses the road and we have an accident after that, we go and blame the black cat because we get fooled by this biological predisposition to see patterns. See the wording here? Humans possess a biological predisposition to see patterns when none exist. And he says we underestimate the role of randomness. So what has that got to do with probiotics? Michael Shermer said the following. He said, the human brain, our own brain, is a belief engine. We are programmed to believe things which are not true. At first forms beliefs from patterns in sensory data, then seeks confirming evidence. We first make a belief and then try to validate it. It should be the other way around, right? We should first look at the data and then believe. Most of us, myself included, we want to believe certain things first and then go back and read to confirm it. So that is what the Believing Brain, the book, is about. Food. We blame food for everything. We blame food for heart disease, for strokes, for cancer, for whatever, dementia, being short, tall, beautiful, ugly, whatever it is. Everything, food takes the blame. Why? Because food is visible, it is popular, it is easy to blame, it is tangible. What if food wasn't such a big element in disease? Maybe many years later, people are going to find that. Maybe there's something else. Maybe we are inhaling chemicals through our air, which are easily getting into our system, but we can't see these chemicals. We blame the food. Now let's go into some statistics. Those of you who follow cricket will know that India made the remarkable feat of losing 20 tosses in a row. Do you know the probability of doing that? The mathematical probability is a coin has two sides, right? It should be 50 and 50, or 1 by 2. But India made it 1:1 million. So it is 1 out of 1,000,000 times you get that combination. Now why is that important? Let's look at a probiotic being used to cure a disease. Let's say IBS, my area. A group of people, this group gets probiotic, that group gets placebo. Everybody has IBS. Let's look at the outcome. I am saying I'm giving this probiotic because it does not work. Let's assume this probiotic does not work. It's an ineffective agent. So ineffective probiotic here, placebo there. Do you think the results will be equal? No. There will be a difference. And guess what I will, as an investigator, say? Yeah, my probiotic group had less headache, so this group is better. Those guys had a little bit more diarrhea than these guys, so probiotic is better. That's what I said. We look for patterns to confirm a belief. So this exists in medical literature. So if equal outcomes are expected, this is the RTK I mentioned. What if both arms showed different outcomes? What if the treatment group showed a better outcome? Is that because of the treatment? No, it is just randomness. It is just a random event. But these random events get celebrated as successful clinical trial. And then they say, oh, the p-value is less than 0.05. Oh, everything's fine. Everything's on the p-value. Now I'll just take a minute to explain the p-value. A p-value of less than 0.05 does not mean there's a 95% chance this drug will work. In statistics, it means completely different. It just says, if we're assuming that the drugs are the same, there's no effect at all. Suppose I repeat the study 20 times, one time I'll get a false positive result. That's all p-value says. So we accept that as acceptable, that's all. It doesn't talk about accuracy or scientific validity or it simply says if you repeat it, 1 out of 20 you'll get it wrong. So with that, let's look at acromantia. This is one of the most, let's say, talked about probiotics. They tried this as an RCT, this group from Europe. They found that the treatment group had better insulin sensitivity and so on. But the number of participants was 32. Do you think that's valid? What I found curious was, for such a remarkable outcome, in 6 years, no other study was done in the same format. If it is such a remarkable finding, why was it not replicated? So that's a natural question. So be careful. All I'm saying is, be careful when you read such studies. Just because it's written, just because it's RCT doesn't mean it's the truth. It has to be replicated. The side effect that we ignore is called financial toxicity. We say probiotic is safe, so why not try it? No, there is financial toxicity. What is financial toxicity? You do the math, 15,000 rupees a year for daily use. And that takes us to what is called marginal utility of money, because If you take a sum of money, let's say 5,000 rupees, say the yearly expense, we think, come on, it's 5,000 rupees. After all, it's for treatment, that patient's going to feel better. But I just want to assert that rupees 5,000 means different things to different people. So, in the interest of time, I will just skip that section. There's a little bit of economics I've built into this talk. We don't have time for that. But basically, it says this. If you give 5,000 rupees to a rich man, for him it's just an extra shirt. But for a poor person, it's a month's supply of grocery. A whole bit of difference. The same amount of money. So that's called discretionary income. We don't have time to discuss that. Basically, it says what is left at the end of the month is discretionary income. A poor person has very little discretionary income compared to wealthier people. So the ratio of the two, if you give them both 5,000 rupees, the rich man may see it as 5,000, but a poor person could see it as even 5 lakh rupees. Because the ratio of the discretionary income to the poor person has very little money at the end of the month. So if you do the math, if you see the math here, it's called a discretionary income ratio. What is left behind for the rich man is 10 lakh, and what is left behind for the poor man is 10,000. So the ratio is 1 in 100. So 5,000 for the rich man is seen as 5 lakh for the poor person. So even if it's 100 rupees, be very careful, is my message. So here's our paper on catastrophic medical expenses. You can look that up. I'll send it to you. And it goes in line with our principle, first do no harm. So now coming to ophthalmology. Uveitis, among many other eye conditions, has been linked with probiotics or difference in gut microbiome. Is it triggered? Here's the mechanism. If you look at the picture, the same mechanism I showed you, the T regulatory cells, which are the policemen. These are the policemen, the T regulatory cells. Once the policemen go on leave, the aggressive T cells take over. And these aggressive T cells will travel all the way to the eye, which is basically an immune privileged organ. And they can disrupt the blood-retinal barrier and by retina-specific T cells from the periphery. So that is the proposed mechanism of autoimmune uveitis, one of the mechanisms. Now these are my notes on immunology. This is all of immunology on one big piece of paper. And from that, I'll show you just the T helper cells. That's the location. I've shown you the location of T helper cells there. So very briefly, if you take a naive T cell, it can differentiate into the good policeman, that's the T regulatory cells. They are the peacemakers. They stop inflammation. Whereas these guys are troublemakers. They work in certain settings, but if you let them loose, they can cause trouble. Say, for example, the Th17 cells can travel into the eye and break the blood-retinal barrier, secrete IL-17, and recruit neutrophils. The hypopion that we see as ophthalmologists is believed to be this Th17 mechanism. Whether they are linked to gut bacteria, that is speculation. All that is speculation here. So I'm not saying this is a fact, but this is a hypothesis. I'll say this. When we look at a chessboard, we may think a rook is important. But having it alone does not necessarily win the game, because so many other players on the chessboard, the ability, the other key, the opponent, so many other factors decide the outcome. So just because we find something, it doesn't necessarily mean it is a big deal. That applies to immunology, gut bacteria, gastroenterology, I'm sure to ophthalmology as well. And there's a study from India that shows that Prevotella, a bacteria, was found more in the gut of Indian people with idiopathic or autoimmune uveitis, come from Hyderabad. The interesting thing about Prevotella is that it is seen in rice eating and high carb diet populations, which India is known for. So it's something that you might want to look up in the future as well. If somebody else can replicate Dr. Chakrabarty's study, that will be interesting. When women are pregnant, they're often told, you need to eat for 2 people. I'm sure all the women here have heard that. But think of it. All of us need to eat for 2 people. One for me, and the other for my gut bacteria. So eat those plants and the vitamin, and your gut bacteria most likely will be happy. Otherwise, you may come and see me. So we can change our gut microbiome by changing our diet, for better or for worse. Here's a good diet. I don't have to explain, the pictures say all. The bad ones. Now, I've got nothing against hamburgers, they are the poster child, but the samosas and the chaats, everything will be in this corner. So there's no Western bias here. Much of the evening snacks we eat in Kerala fall into Undamburi. All these things happen here. They all worsen the microbiome. Undamburi is the bestseller in our canteen. And if you thought having a diet beverage is healthy, think again. I've stopped drinking diet beverages after this. They're not good at all. There are studies which I have decoded on national media for why these diet artificial sugars can increase your risk of diabetes. Imagine, you're not a diabetic. You want to cut your sugar, you take these artificial sweeteners and you become a diabetic. How ironic is that? It happens, and this is the mechanism. Now, to answer your question, you asked me, should I write a probiotic for my patient? I'll tell you they don't work. I'll tell you why they don't work. There's something called colonization resistance. The existing gut bacteria are not going to give up they're like politicians. They're not going to give their chairs up easily. They'll hang on to their chairs. They will not get up. The new bacteria can come in, they will not be let in. So you may be thinking you're taking this expensive pill, but the pill just makes expensive feces, that's all. It does not really cause any change in your gut, trust me. The second is, when 2 people take the same probiotic, it may work for 1 person. You know why? Because if we take it along with a good diet, it works better because the bacteria needs fiber. And the third is, many of these pills that we get from the medical store, they don't really represent the true natural bacteria. They are what they can commercially produce successfully, but they don't represent the normal gut flora. Gut flora has hundreds of species. These guys are selling 1 or 2 species. I'm not belittling the work, I'm saying it is not as big as we think it is. It's not like suddenly you take lactobacillus, tomorrow your gut is full of lactobacillus. It doesn't work like that. Guaranteed. Now, this is really, really bad news for you. Most doctors, including me, had the habit of, if I ever write an antibiotic, I'll give a probiotic. No thing, it's like medulla level thinking. Cerebrum is shut off. Antibiotic, probiotic combination. Chalo, here's prescription. No. Here's a paper. I've decoded this on Twitter. That paper came out, I think it's from one of the basic science papers. Maybe Cell. I'll give you the main message. Suppose you give probiotics after an antibiotic. The probiotic may not necessarily help the patient. You know why? I'll put it like this. Suppose I give an antibiotic and all these people go away, this section. So all the seeds become empty, and I throw my probiotic there, those seeds are taken. Now when those guys return, there's no chair left. The probiotic is sitting there. Remember, the normal gut flora is more healthy than your probiotic. So my antibiotic is destroying these guys. And I'm putting probiotic on their chair, so they will find it more difficult to come back and recolonize. I hope that makes sense. So paradoxically, we may be prolonging their recovery or delaying their recovery. So that's a thought for you. This is one paper, as I said, more people have to look at this phenomenon. So it is not as simple as 1 + 1 = 2. So the key points from my talk is 1, microbiota. I would say there's still a mystery. All these are theories. I can make a theory and say, if I throw a stone very hard, I can reach the moon. Yeah, theoretically that is true, but does it happen? Probably not. I can say, yeah, theoretically, yeah, it's mechanistically possible. If I throw a stone really hard, it should reach the moon. If it reaches escape velocity, it can escape the Earth's gravity. Yeah, it can reach the moon. Does it happen? No. So many of the claims on these lines are based on mechanistic possibilities. So I'll caution you about that. Commercial pills have limitations for the reasons I mentioned. And the other thing is the commercial pills are all oxygen-friendly pills. See, you can't take an anaerobic bacteria and pop it, because the moment it gets exposed to oxygen, they die. So where are your colon bacteria? So you're just giving them small bowel bacteria, which is again one more reason they don't really work. So I already talked about the diet, so I'll skip that. And my last slide is this. When we see social media pundits, some of them wear white coats also, they push probiotics and diets, and here's the key. If any of them seem 100% certain about something, just walk away. And remember this. The problem with the world is that the intelligent people are full of doubts, while the stupid ones are full of confidence. I thank you for listening.
Thank you, sir, for that insightful talk. The real test begins for many of us